Thank you, Dr. Spear, for the kind introduction. It's a great pleasure to be here to tell you the story about our, we call the self-destructing salmonella vaccine delivery system, and how we teach those bacteria new tricks to fight cancer. So our lab has a long-standing interest to develop vaccines against the infectious diseases. So I will use a couple slides to explain what brings us to the field of cancer therapy. So this is the number from, according to American Lung Cancer Association. So unfortunately, about 300,000 Americans may die from lung and the other three most common cancers in 2014. So cancer is still one of the leading causes of death in the world. So this cartoon shows the microenvironment in the tumor. So because the abnormal development of blood vessels in tumor, so inside of tumor is a hypoxic condition. So this makes it very difficult to send the drugs in this area, and also oxygen is important, required for radiation treatment. So tumor hypoxia is a major hazard of cancer therapy. So we see the need of development of novel therapies targeting hypoxic cancer cells. Bacteria has been investigated as anticancer agent. So those bacteria has one unique features in common, because they can survive in hypoxic condition, and also they can replicate inside, inside of tumors. And those bacteria has been used as either live, attenuated, or genetically modified forms. Also has been used as a vector to deliver anti-tumor agents. And also the toxin has been used to treat tumors. Most famous is currently published the result uses spore from Clostridium treated cancer. And the positive result not only show in mice, dogs, and also human patients. So among those bacteria, Salmonella is the popular one for a couple reasons. So first, the Salmonella can survive in both conditions, oxidative condition and also anaerobic condition. And also Salmonella prefer accumulate in tumor compared with normal tissues. And most importantly, Salmonella, so they can invade large solid tumors, and also they can invade and affect metastases. So we have developed this, we call the self-destructing Salmonella for antigen delivery and also for DNA vaccine delivery. Those Salmonella, they can lyse in vivo by programmed regulated lysis mechanism. So I will walk you through, explain how this system works. Because Salmonella has very simple life cycles. They can grow, divide, grow, divide. So during this process, newly synthesized bacterial cell wall is required. So if we can control bacterial cell wall synthesis, we can control Salmonella's fate, is life or death. And we want to make Salmonella death because lysis. So if they can lyse in vivo, they can release all the cell contents inside of the organs. So this cartoon shows how we built this system. So we have two components. One is bacteria, the other one is plasmid. So we knock out those genes from bacterial chromosome. So they respond for synthesis of bacterial cell wall And we put those genes into a plasmid. And those gene expression is under control of regulatable promoter. It's an RCP-BET promoter. So in vitro, we can provide a arabinose that's induced for the promoter. They can synthesize the bacterial cell wall. But in vivo, free arabinose is not available. So bacteria will stop synthesizing those cell walls and they will lyse. So this strain, if they want to survive, they have to host our plasmid. And also we can use this plasmid to synthesize protective antigens. So I will skip the details. We have all other tricks just to make sure bacteria they can lyse completely. So this cartoon is a summary of what's happened during the invasion and after colonization. So you can see Salmonella invade to the cells, and they will hide inside of endosome structure to avoid the bactericidal material. And we can control where Salmonella will lyse, either inside of the endosome, or we can make them escape from the endosome. Eventually, those Salmonella will lyse and release the protective antigen. So if you look at the lower panel, you can see this system, if we provide arabinose, they can perfect grow. But if we withdraw arabinose from the medium, they all lyse. And also you can look at our paper for more details. So we improved this system for DNA vaccine delivery. It's for the plasmid part, it's much easier. The only things we did is So we improved this. It's hard. So we improved this plasmid by introduce some DNA nuclear targeting sequence, DTS1 and 2. Because this is a bacterial-bound plasmid, they don't have mechanism to cross the nuclear membrane, so that's why we added those little pieces. And you can see this is improved plasmid compared with the first generation, the expression level of reporter genes significantly increased. So for the strains, it's much harder. So first things, all those, so you can ignore those features. It's just a safety feature for the vaccines. And the first things we need to do for DNA vaccine delivery, we have to make those Salmonella inside of the cells. But I need to learn how to use this one. So after invasion, Salmonella stop synthesizing those invasive genes, and they rather stay outside of the cells. And for DNA vaccine delivery, we can make them into the cell, and they lyse inside of cytoplasmic. They can release those DNA vector. So we made those hyper-invasive strains even after invasion. Out into the host, they continue synthesize those invasion genes. So second things we have to do, because Salmonella cause apoptosis or peroptosis. So they kill the host before the vector can go anywhere. So what we did, we knock out those genes and reduce Salmonella-caused apoptosis, and just give bacteria time to replicate and lyse and release those plasmid. And the last things we did is introduce CFA mutation, because Salmonella will hide inside of those endosomes, so we have to make Salmonella escape from those structures, and they can lyse inside of cytosome. So use this system, we already successfully developed the anti-influenza vaccines. You also can see the detailed information in our publications. So now we can control where, if those bacteria will lyse, and when they will lyse, and where they will lyse. So use this system, we already developed different vaccines, target different disease. And we improve those strains by, depends which kind of disease we are targeting and which kind of dominant protective immunity we want to induce. So what we can use this system for cancer therapy, and which kind of trick we have to teach Salmonella in the other world, which kind of genetically modification is required for those strains are able to treat cancers. So first things we can think about is enhance the ability of those strains induce innate immune responses. And also we can reduce the toxicity and keep the efficacy for tumor treatment. And also we can help those Salmonella escape from the vacuolar structure, and they can release those cell contents or anti-tumor agents to the cytoplasmic. And also, one thing is very important because Salmonella can go anywhere. So we want they target the tumor. And how we can help those Salmonella, they prefer colonize in tumor, but we want they selectively colonize in tumor. So a lot of tricks, I just want to show you a couple examples. So first example is there has some evidence tumor can release a lot of different small molecules. Some of the molecules can attract Salmonella to the tumor. So currently has some argument about those results. So what we did, we play with a couple chemoreceptors just to make them more attracted by tumor. So 3 receptors we have been touched. One is TAR, it's for the acetate amino acid signal. And the other one is TSR, the signal is serine. And so by improve those chemoreceptors, we made those bacteria more attracted by the chemoattractants. And also, I will show you some results from those modified strains. So the other things, the tricks we teach bacteria to learn is, although they prefer accumulating in tumor, but still a lot of bacteria you can find in normal tissues. So if we want to deliver anti-tumor reagent, so we don't want they colonize in normal tissue, so we selected multiple mutations. So we introduced some mutation into Salmonella strains. And what you can see is the ratio of Salmonella they colonize in tumor versus spleen. The ratio has been significantly increased. So wild type at the beginning, you can see, so this is the intratumor injection of Salmonella. So you can see the more Salmonella you can find in the tumor. But very soon, at day 3, you can see they evenly distribute to the normal organ and the tumor. But if with those mutations, you can see they prefer stay in the tumor other than the normal tissues. So we use those tumor models, and those tumor models created by those luciferase labeled tumor cell lines. So our first target is colon cancer. So we use those sub-Q tumor model to select the strains. So you can see this is just one example. Those strains has all the different mutation. The first 2 is for the cell lysis feature, and all others is for the safety feature, or it's reduced endotoxin, or some feature is just help bacteria completely lyse. And the last 3, So last three mutations you can see is enhance the chemoreceptors. So use those strands. This is the improved DNA vaccine vector. And use those strands, they can stop tumor growth. But if we want tumor disappear, we need other, deliver other anti-cancer agents, such as some death ligand. So, and also at the beginning, the idea is very simple. We just want to use our lysis system and make we call the bio-time bomb. So if they can colonize in tumor, if we pack some anti-tumor killing material, and at some time point, they will lyse release those material and kill tumors. So that's all I want to show you today. And at the end, I want to acknowledge my teams. So this is the team who was working on developing a program that regulates the lysis system. And the funding is from NIH and also from USDA. And this is our anti-cancer teams. So Dr. Curtis, who is my long-term mentor and collaborator, he's the one make me believe Salmonella can do anything. So, and this is my team for anti-cancer therapeutic strain development. And also thank you the funding from NIH and CI. Thank you for your attention. I'll be happy to answer your questions.
Any questions?
Do you plan to use Typhimurium for your future human trials, or will you switch straight at that point?
So we prefer use Typhimurium for various reasons, because Typhimurium is a pathogen for mice, and typhi is a pathogen for human. From safety point of view, and also human may pre-exposed to typhi may against our strains. So we prefer the first trial we want to use Typhimurium other than typhi.
Yes, sir.
I have a similar question to you. Why don't you can use the enteritis, Salmonella Typhimurium for the invasion?
Can you repeat the question?
As you use the Salmonella Typhimurium in order to invade in the colon cancer tissue. So can we use the Salmonella enteritis for the better invasion?
He's asking the question, basically, different salmonella invade different tissues. So can you basically change the species of salmonella you use and then get a cancer in a different tissue?
Yes, that's a very good question. So because my first funding is on the colon cancer, so that's why I chose Typhimurium. They will invade orally through across the gut, is close to the tumor site. So if for target other, yes, people choose different salmonella strains target different cancers.
But you can go further, I'm sure, and use streptococci or pneumococci to do a lung system, because the system you use for killing the bacterium is very much like the adenovirus system that people use for ferrying genes into cells that are you have competent cells and you have incompetent cells. So you have the viral vector, which is not a replicating vector that you can put into So presumably you can use streptococci for lungs and different bacteria for different parts of the body. Am I right in thinking this?
Yes, that's a very good point. And also, if we were thinking if we target some brain tumors, we should choose the bacteria. They can colonize in the brain. So that's very good thing.
The other thing is, of course, is that with this method of attenuation, if you like, you've basically got the possibility of vaccines, which, irrespective of whether a person has cancer or not, I mean, they can also be used as a prophylactic vaccine, can they not?
Yes, so actually, the major job in our lab is to develop vaccines against the infected disease. I show you a couple of papers. We already successfully used lysis system deliver vaccine against Yersinia, Clostridia, and pneumonia, and influenza. Use antigen delivery system and also the vaccine system. And also currently we were successful with some funding from company to develop anti-agriculture important parasites, and the result was very excited. So the paper is on the way. So just to use this, we happened to get we were thinking maybe convince human drink salmonella, it's very difficult duty. But if cancer patient, if I will die tomorrow, I may be willing to drink those salmonella. That's the first idea to use.